It's my privilege to be currently the president of the Oligonucleotide Therapeutic Society. I'm Annemieke Aarsma-Rus, and I would like to welcome you to our first virtual conference. And I went already to a couple of the networking sessions, and I know that we have some returning attendants, but we also have a lot of first-timers. So warm welcome to all of you. And for the newcomers, I would like to very briefly touch on the mission of the Oligonucleotide Therapeutic Society. So what we want to do, we want to foster academia and industry-based research on the development of oligonucleotide therapeutics. And we also want to foster young researchers. Because we are a new field, we are aware that we really need to have an open dialogue on the science to move the science forward, but also we need to prepare for the future. So we really would like to invest in the young researchers who will need to carry along the new field when we retire. So we have a couple of tools to achieve our mission, and well, the one that you're currently attending is the meeting. We generally have in-person meetings. This year, of course, we had to do a virtual meeting where we really want to showcase high quality science. And we also want to give the junior members a platform. So if you've not already done so, please have a look at the posters, the on-demand short talks. Please have a look there as well. And also use the opportunities to digitally interact with all these people. In addition, we also have educational webinars, and these are all recorded and available on our website. There's a lot of other information on our website as well. If you want to go there, please scan the QR code. We're also on Twitter, so if you can see already a lot of tweets on the hashtag OTS Virtual, and you can follow us on OT Society. And we have a journal that is aligned with our society. So these are our current board members. So we have a number of expert board members. We also have 4 trainee representatives. And normally this year we would have new nominations. But because of these exceptional times and to ensure the continuity of the society, all board members unanimously agreed that we should extend the term by 1 year. So everyone agreed to stay on for 1 year, so I appreciate everyone's dedication. So there will be no voting for new board members this year. There will be voting next year, and then the next term will start in 2022. And we felt this was better to ensure the continuity of the society in these exceptional times. There is one exception, and that is the trainee representatives. The current trainee representatives will stay on 1 more year, but there will be vacancies for probably 2 more trainee representative board posts this year, and that will be announced later this year. So if you're a young researcher and you would be willing to become a trainee representative, stay tuned, because there will be more information about this later this year. So currently the status is that we as a society have 661 members, and as of 10 minutes ago we had 1,029 attendees, and this is really a record number. So we really appreciate that everyone was willing to come to our virtual meetings. And also we're a growing field. We have 3 more approved oligonucleotide therapies, and we are now at 11. So I already mentioned the website. Please have a look there. Those of you who yesterday went to the educational session know that these sessions we've been doing them since 2015. They're all recorded. You can find these videos on the website. We also have these regular educational webinars. Also these you can find on the website. And you can find lots of other things on the website. So please have a look. I already mentioned our journal. So the Nucleic Acid Therapeutics is the official journal of OTS. The scope seems self-evident, and also this journal is growing. And you may have heard that our impact factor was 4.8. However, it turns out that the impact factor people miscalculated. So it's actually 5, and this will be formally announced in October, but already give you the scoop. So the impact factor of the journal is now 5, and also the downloads are increasing, while the peer review time is decreasing. So I really would like to invite everyone to submit their oligonucleotide therapeutics related work. As a member, you get a discount for open access fees. And I would like to highlight also the special issue on negative results that will appear early next year. And negative results is, of course, a misnomer. If our hypothesis is wrong, that also advances science. And something that doesn't work can also advance science. So we really also like to invite everyone to keep also sharing with us the things that do not work. And I would like to take this opportunity to thank the co-editor-in-chief, the executive editor, and also the other editors for working on this journal and in improving the impact factor and the quality of the journal. Another big thank you to the sponsors. So when it became clear that we had to transfer from an in-person meeting to a virtual meeting, everyone stayed committed. So we really appreciate this, because the fact that we can offer everyone such a high quality virtual experience is due to this sponsorship. So big thank you. And also thank you to the exhibitors who also stayed committed. And please do have a look also on the meeting website, because these people are all working, these companies are all working in the oligo field, and many of them probably have tools and compounds that you need for your experiments. So then a final big thank you to the people from event innovation. So Jerry, Alexis, and Cindy, and they really went beyond the call of duty to convert our meeting from the in-person meeting, which is a lot of work anyway, but then halfway through planning that, they had to convert it into a virtual meeting. And I think they really did exceptionally well. So I would like to invite everyone to give them a virtual applause, and hopefully they can hear it in California where they're currently at. And then I would like to hand over to our scientific meeting chair, which is John Watt. So please give an applause to Jerry, Alexis, and Cindy.
Well, greetings. It's really good to see you all. Thank you for that thunderous virtual applause. As you know, we were all supposed to be meeting this week in beautiful Montreal in the autumn at the sunrise, as you can see behind me. And instead, we find ourselves meeting here in cyberspace. But it is good to see you all. It's good to see you all virtually and be together. As Annemieke mentioned, the OTS is created to help each other, to advance the field. And so we know that therapeutic development is hard. Therapeutic development in a pandemic, as we've all discovered, is even harder. But the OTS exists to enable us to help and encourage each other on the journey. And during a virtual meeting, we all have to think more about that. Perhaps at an in-person meeting, we have the instinct to share an encouraging word, to make a comment on a talk. And during a virtual meeting, we have to remember that the level of feedback is inherently a bit lower. So please take the time to encourage those who are sharing, suggest an idea for follow-up, ask a good question. Based on the pre-conference sessions yesterday, the educational workshop and the next generation workshop, the level of interaction was very high. So thank you. It seems that you are all already stepping into to doing this. It is now my pleasure to introduce our keynote speaker for the OTS 2020 annual meeting, Judy Lieberman. Judy received her PhD in physics from Rockefeller University and worked at Fermilab for a while before completing an MD from Harvard Medical School and MIT. After stints at Princeton and Tufts, she moved to Harvard Medical School and Boston Children's Hospital, where she's currently professor of pediatrics and endowed chair in cellular and molecular medicine. Judy is an immunologist and has carried out research on both basic and translational aspects of the biology of cytotoxic lymphocytes. RNA interference has been another theme of Judy's research for nearly 20 years. Her group was the first to show that RNAi could be the basis of therapy in an animal model. And she has explored methods for targeting small RNAs into specific cell types in vivo. Judy's scholarship has garnered many awards and honors, far too many for me to list here, but including recently, last year, in fact, her election to the National Academy of Sciences. So Judy, I'll hand over to you. Thank you for being here with us, and welcome.
You have to stop your screen so I can share mine.
I think I did. Yeah. Go ahead now. Try again now.
Okay, great. Okay, thank you, Anna, Miki, and John for this very kind invitation to give the keynote talk of this lecture. It's a true honor. I decided that I was gonna, since I was sort of present at the beginning of this journey, I would sort of structure my talk around the development of RNAi therapeutics, which has been very exciting to participate in. So it's just, it took 20 years from the discovery of RNAi and siRNAs by Fire and Mello in 1998 till the first RNAi-based therapeutic agent developed by Alnylam, Fas-siRNA, was approved by the FDA just 2 years ago. And now we're really in a period where the fruits of all this research and labor are going to be realized, and as more and more drugs enter clinical use. I was very fortunate to participate in some of the important research that set the stage for drug development. Just very shortly after Tom Tuchel showed that RNAi worked in mammalian cells and that you could knock down gene expression selectively, one gene at a time. In 2001, I was approached by a postdoc then in Phil Sharp's lab before the paper came out, asking me if I thought it might be possible to get RNAi to work against HIV. And that's sort of, the rest is history. We're able to do it in vitro in primary cells. We could knock down either viral genes or host genes like CD4 and CCR5 that are needed for the virus to replicate. And very shortly thereafter, we showed that we could get RNAi to work in vivo in models of autoimmune hepatitis, where it was basically life and death, and also later on developed the first targeted gene silencing for RNAi therapeutics that was mediated by antibody binding to receptors. I became aware of RNAi by reading the Fire and Mello paper in 1998 in worms. For me, it was a very exciting paper, and I kept asking my friends in the C. elegans field, how does this work? What's going on? And then when Tom Tuchel and Sadie El-Bashir showed that it worked in mammalian cells, that was sort of the impetus for really seeing if it could be harnessed for immune therapy. And that was in 2001. It was the beginning of Space Odyssey. and so the idea that was put forth in 2001, we actually didn't know anything about the mechanism of RNA interference, and we didn't know, so basically starting to develop therapeutics was really done in the dark before a mechanism was apparent. It soon became clear that if you could somehow get a small double-stranded RNA that was homologous to the sequence in a messenger RNA into the cytoplasm of cells, it would be taken up by the RNA-induced silencing complex, or RISC. One strand, the passenger strand, which was not antisense to that messenger RNA, would be removed and degraded, leaving an unpaired small RNA in the RISC complex, which could then seek out messenger RNAs that were homologous or partially homologous, like in the microRNA pathway. If the homology was good, then the active component, Argonaut component of the RISC complex could cleave the mRNA, which would then leave the complex, get degraded, and you get a recycled RISC that was ready to cleave another molecule. And basically, because it's catalytic, it's incredibly potent gene knockdown. Fewer than 1,000 cells, siRNAs in the cytoplasm, can cause complete knockdown. And now with chemical modifications that enhance the stability of the siRNA, can last for as long as 6 months. So my postdoc, Erwei Song, had the idea of trying a hydrodynamic injection, which Mark Kay had shown could be used for efficient hepatocyte uptake of small RNAs. And hydrodynamic injection is a sort of scary process where you inject a mil into a mouse that has a blood volume of only 5 mils very rapidly, as fast as you can. And that kind of injection can, in some cases, kill the mouse, but if not, you can get very efficient uptake of fluorescent Cy5 conjugated siRNAs into hepatocytes labeled with albumin, here close to 90%. We decided to knock down Fas, which is an immune activating gene that causes cell death in the liver, recruits lymphocytes that then start attacking and killing liver cells. And so here in this RNase protection assay, we could show a rather selective gene knockdown of Fas, but not any other genes that we looked at in the death receptor pathway. And even though gene knockdown at that time was not, with naked RNAs, wasn't completely selected, very rapidly it was found that chemical modifications of the siRNA could make it virtually selected for one gene. And the knockdown lasted for 10 days post-injection, which was a lot longer than anyone would have thought. And the therapeutic results were really amazing. We used a very fulminant model of hepatitis, where you inject Fas antibody into the belly of mice, and all the mice die within a few days. However, in mice that receive Fas siRNAs but not control siRNAs, about 80% of the mice survived. So this really sparked incredible interest from biotech and pharma to try to develop therapeutics for that harness RNA interference. And because our initial interest was actually seeing if we could use RNAi to suppress viral infection. It took us quite a while to get to successfully do that for HIV because it infects lymphocytes that are very hard to transfect, even in vitro. But we were able to show that siRNAs against 2 viral genes could, if applied topically to the vaginas of mice, could protect against sexual transmission of herpes simplex. In this graph, in purple are mice that had no symptoms, and in red, mice that die. And you can see that almost all the mice at this challenge dose died in the control groups, but with the siRNAs, they were largely protected. And we also showed that if we knocked down the receptor for the virus, we got similar effects. People were very excited about RNAi therapeutics because you can get very specific gene knockdown. Later on, it was found you could actually target gene knockdown to specific cells. You could knock down any gene, greatly expanding the universe of druggable targets. With chemical modifications, the RNAs themselves are not immunogenic and not toxic, although some of the delivery mechanisms are. And once you know a target that's responsible for a disease, you can actually very rapidly identify sequences that work and do animal testing and be ready for applying for an IND within a year or so. The drugs we have now can last in non-dividing cells for months, many months, and can be given subcutaneously. So the biggest challenge for RNA delivery, for using RNA as a drug, was getting the siRNAs across the cell membrane, and once endocytosed into endosomes, out of the endosome into the cytosol, where the risk complex resides. And these two roadblocks, the biggest one was getting across the cell membrane, but getting out of the endosome is also a pretty formidable challenge. Took up the most research effort to develop RNA into drugs. And we, you know, my strategy and that of Alnylam, which I was associated with for many years, was that if you can understand these roadblocks and really seriously address them, you could actually overcome them and develop drugs. And one of the methods that Anders, which up in my lab, developed was a way to visualize the release of RNA from endosomes into the cytosol and to coordinate it with gene knockdown. And basically, he found that gene knockdown actually occurs within complete gene knockdown within a few hours of uptake and release, that RNAs that were complexed in lipid nanoparticles like were used for Fas-siRNA were released at a very specific time early on after endocytosis. He was able to quantify how many RNAs were needed for efficient knockdown and show that even for what's a clinically effective drug like Fas-siRNA, only a few % of the RNAs that are endocytosed are indeed active. So, so there are many ways to try to deliver RNA or other oligonucleotides into cells. It's easier to get antisense oligos, which are single-stranded, into cells than double-stranded siRNAs. But the first efforts in the field centered on using lipid nanoparticles. And that is was the basis of the first drug that was approved, which was Fas-siRNA, developed by Alnylam to treat a rare, rapidly progressive and fatal disease called transthyretin-induced amyloidosis, which is caused by mutations in transthyretin, which is a liver synthesized gene. And it doesn't fold properly and causes large fibers to deposit in cells, causing the disease, and that's called amyloidosis. And it affects mostly the central nervous system and heart. And basically, lipid nanoparticles work pretty much exclusively for knockdown in the liver. And because the liver is the factory of the body that manufactures a lot of the blood proteins, it was a place where gene knockdown could have effect against multiple diseases. So this is a cartoon of the phase 3 trial in which patients were randomized to receive the encapsulated siRNA at a low dose intravenously every 3 weeks, or placebo, and analyzed for changes in their neuropathy score. And basically the results of this, and actually many of the clinical trials for testing siRNA, are amazing. Here, I've never seen p-values like this, 10 to the minus 24th, for clinical trials. All the secondary endpoints were met, and the drugs were reasonably safe, although there were reactions to the injection site and the liposomes. And patients who were basically wheelchair bound from the neurologic disease were some of the patients able to return to a full working life. However, lipid nanoparticles, although they work, are not ideal. There were immune responses. They're very complicated to manufacture and cause clinical toxicity. So it would be important to try to get beyond lipid nanoparticles. And Nataya Manoharan from Alnylam basically developed conjugated siRNAs conjugated to sugar, that GalNAc, which is recognized by a highly abundant receptor on hepatocytes specifically. And by conjugating the siRNA and doing a lot of chemical modification of the RNA, he was able to develop a very effective way to get gene knockdown in the liver with almost no toxicity. And the drug could be given subcutaneously. And basically, the GalNAc binds to the asialoglycoprotein receptor, which is present in hundreds of thousands of copies on the surface of hepatocytes, gets taken up. Some of it is released through a mysterious, not well understood process, but some of it can get of the siRNA that isn't released can actually get goes into recycling endosomes that can then be a storage for the RNA and probably account for the long durability of action. And so the second drug that was approved was a drug called Fas-siRNA, also by Alnylam, to treat another set of genetic diseases, the porphyrias. And these are genetic mutations, also very rare, in the enzymes that cause heme bias, that are responsible for biosynthesizing heme that's involved in making hemoglobin. And these patients have episodes of severe pain and can develop chronic renal and kidney and liver disease. So Fas-siRNA is based on the siRNA to the first enzyme in heme biosynthesis, aminolevulinic acid synthase. As I said, it's given sub-Q. It's given monthly in this earlier version of the drug. The results, again, are pretty dramatic in reducing the painful, debilitating attacks of the disease, with, again, very little in the way of safety concerns. And the results have been validated and sustained in open label extension studies. And then, so this method of GalNAc conjugation has become basically the basis for a number of other drugs, and has replaced using lipid nanoparticles. And there were 2 drugs in which phase 3 trials have been completed with amazing data, which are likely to be approved in the next, within the next year. One is to treat hypercholesterolemia, which affects a lot of people as both a genetic and idiopathic disease. The target is an enzyme, PCSK9, that regulates the recycling of the LDL receptor that picks up cholesterol-laden LDL particles. The siRNA is a GalNAc conjugated stabilized siRNA that's administered only every 6 months, which is pretty amazing. It was developed by Alnylam with the medicines company, which was bought by Novartis earlier this year for a lot of money. And it has a very good safety profile. And basically, in the 2 phase 3 trials that have been reported involving many thousands of patients, the reduction in LDL cholesterol was about 50% in both trials. And then the final drug that's likely to be approved also within the year is to treat a disease that's mostly in children that causes large kidney stones made of oxalate, and that's rapidly progressive, leading to kidney failure and requires liver transplant and kidney transplant to treat. And this clinical trial is not published, but the primary endpoint was the reduction in oxalate in the urine. And again, the results were pretty amazing with monthly loading doses and then every 3 months treatment. So we're really on the brink of a new epic in RNA therapeutics. Oops, sorry. So Alnylam, which I was associated with for a long time, has a pretty ambitious pipeline of drugs that are being developed and evaluated in the clinic. And they're really moving from genetic diseases like amyloidosis and porphyria and hyperoxaluria into more common diseases and infectious diseases like hypertension, kidney, liver, hepatitis, non-alcoholic liver disease, and very recently, potentially to COVID-19. I think the next frontier is going to be going beyond the liver. They have unpublished data suggesting good delivery and gene knockdown in the central nervous system and eye, and even the possibility of potentially an oral drug. So what the rest of my talk, I'd like to focus on how do you get RNAi therapeutics to work beyond the liver, because that's something I've really focused on in my lab. And recently there have been a paper by Anastasia Kurvova suggesting that ligating different lipids to the siRNA can target them to the central nervous system and other tissues. I want to point out that delivery into cells is not necessarily equivalent to gene knockdown, and both have to be shown and efficient for a drug to work. And there are also the unpublished efforts and undescribed efforts of Alnylam. There's also efforts to develop targeted lipid nanoparticles for tissues outside the liver. And in particular, I'm a fan of the work of Dan Peer, who has developed a very robust way of antibody-targeted RNA therapeutics with lipid nanoparticles. And what my lab has been focused on is adapting a platform that was developed by Paloma Giangrande and James McNamara, Ellie Gilboa, and Bruce Solinger, and also adapted by John Rossi, that uses aptamers or structured pieces of RNA or DNA for targeted gene delivery. And that's what I'm going to spend the time talking about. So Paloma was able to link an aptamer, which you can think of as a nucleic acid antibody, via a linker to a siRNA. And this is a platform that I think is very attractive. Basically, it's a single piece of RNA. RNA is very non-immunogenic. It's simple to manufacture. By changing the aptamer, you can change the targeting, what cells get knocked down. And by changing the siRNA, you can knock down virtually any gene, or even introduce microRNAs or other types of RNA into cells. We've found that it can be given sub-Q and gets selectively to the cell tissues that express the target of the aptamer and cause gene knockdown selectively and safely. And we chose for our research to look at cells that are very hard to transfect, lymphocytes, because we're interested in HIV and cancer cells, which are disseminated throughout the body. And I'll just show a few slides of our work with CD4 aptamers to deliver siRNAs to block HIV transmission. And this is the work of Lee Wheeler. And we took an aptamer that was identified in the 90s. It's very long. You have to in vitro transcribe it, and linked it to a siRNA. And we're able to show in primary macrophages, which are very difficult to transfect, that we could completely suppress HIV infection by knocking down viral genes like GAG and BIF or CCR5, the co-receptor or combinations of them, which is shown here. Oops. I'm sorry. And basically here we're labeling, we're infecting cells with HIV. We're introducing either CD4 aptamer linked siRNAs or a control prostate specific antigen aptamer, and looking at HIV replication in green by FISH and siRNA introduction in red with the fluorescent siRNA. What you can see is that this is the control infection. The PSMA control doesn't work, but the CD4 aptamer linked siRNA, and we call these ASICs for aptamer siRNA chimeras, basically knocked down completely or prevented HIV infection and got into all the cells. And we could deliver these siRNAs, ASICs, intravaginally into humanized mice that can be infected with HIV. And here you can see that the uptake, the RNA gets taken up into macrophages, which very dimly express CD4 or CD4 lymphocytes, but not into CD8 lymphocytes. So the delivery is very specific, and the knockdown was efficient at just injecting picomoles of siRNA into mice. And again, it was specific, looking at two different genes, CD45 and CCR5. And the results were pretty dramatic without any optimization. We could inject siRNAs that targeted CCR5 and viral genes, just CCR5 or just viral genes, and completely prevent HIV infection. Here we're looking at the decline in CD4 T cells or the upregulation of the HIV P24 antigen or HIV RNA in the blood. And in all of these mice, there was no infection. However, I could never get anyone to develop this clinically. And then we moved on to look at cancer, and the rest of my talk will focus on our efforts in cancer. So we found an aptamer to EpCAM, only 19 nucleotides in the literature by this group. It binds with low nanomolar affinity to EpCAM, which was the first tumor antigen described. And I liked EpCAM because it's only expressed at very low levels and probably hidden at gap junctions in normal epithelia, but highly expressed in all the epithelial cancers. And it marks the really malignant subgroup of cancer cells called cancer stem cells that are not treated with chemotherapy and are responsible for tumor relapse and metastasis. And in our first paper, which was 5 years ago, we, as a proof of principle, targeted PLK1, which is the kinase required for cell division. And it turned out that PLK1 was the only kinase that turned up in a screen for cancer stem cells. So it was fortuitous. And basically, the EpCAM targeted aptamer siRNA chimeras, or ASICs, get into epithelial tumor cells, here shown a mouse triple-negative breast cancer and a human triple-negative breast cancer, and has similar affinity for both mouse and human receptors, but not into a fibroblast, which doesn't express EpCAM. And the knockdown didn't happen in the cells that are tumors that are mesenchymal or EpCAM negative, but it happened in a dose-dependent manner in EpCAM positive cells, and was able to kill EpCAM positive cells, but not affect EpCAM negative cells. And basically, if we took the fluorescent ASICs and put it in a petri dish with normal tissue and a tumor slice, we found that the fluorescent RNA, which you can visualize as red, was only taken up by the tumor and not by the normal tissue, and that's shown here. Then we wanted to see if this could block tumor initiation, which is the feature, in vivo feature of cancer stem cells. So we took EpCAM positive triple-negative breast cancer and treated them ex vivo with the EpCAM ASICs or a GFP control, and then looked at whether tumors were started. And as you can see, this was very effective at killing off the cancer stem cells. Moreover, if we implanted a EpCAM positive tumor in one flank of a mouse and EpCAM negative tumor in the other flank, and then injected the fluorescent ASICs into the scruff of the neck, we found that the injected RNA only went to the EpCAM positive tumor and not to the EpCAM negative tumor, and didn't really appear to go very much elsewhere. And this is just quantified here. So we're able to get very selective in vivo gene expression and also knockdown in tumors in vivo through sub-Q injection of ASICs. And using this PLK1 targeted ASICs, we're able to, with a couple of injections, completely eradicate a palpable aggressive triple-negative breast cancer, but not with the controls, and had no effect on the EpCAM negative mesenchymal tumor. So these ASICs provide a very good way of transducing cancer cells and potentially treating them. And we've been able to show that these ASICs also work very effectively for other solid epithelial tumors like prostate and colon cancer. And I'd like to finish up by just describing how we've been using the ASICs as a new approach to cancer immunotherapy. Ellie Gilboa has been really in the forefront of trying to develop methods for using RNAi for tumor immunotherapy. And basically, tumor immunotherapy has really revolutionized cancer treatment in the last few years. However, most tumors, even of the tumor types that respond to therapies, respond to immunotherapy, do not respond. So only a minority of patients benefit. And so we thought maybe we could use ASICs to broaden the scope of immune therapy and get tumor cells that are not visualized by the immune system to be recognized and killed. And so we decided to use the EpCAM ASICs to knock down a bunch of genes that interfere with tumor recognition or killing by multiple mechanisms. And so basically the cancer cell, when it dies, releases antigens that are recognized by antigen presenting cells like dendritic cells that move to the lymph node. They activate T cells to recognize the tumor antigens. Those T cells go back into the tumor and can kill the tumor. So Ying in my lab in a year basically knocked down about 8 different genes that work at different stages in this tumor immunity cycle. And they all seem to work and profoundly increase anti-tumor immunity. And so I'll just show you a few examples. Here we're actually taking a page from Ellie's work in Nature in 2010, where he showed that if you knock down an RNA quality control pathway, nonsense-mediated decay, you could get tumor control. And here we're showing our data with the EpCAM A6 in a aggressive triple-negative breast cancer. We could suppress tumor growth. We could increase the number of infiltrating CD8 T cells and reduce the numbers of inhibitory regulatory T cells. The T cells that were in the tumor downregulated the receptors that are targeted by checkpoint therapy, the inhibitory receptors, and the tumor infiltrating lymphocytes became much more functional. They released their cytotoxic granules, they killed tumor cells and produced cytokines that are active against the tumor. Then we thought that if we inhibited DNA repair, the tumors would express new antigens. So we knocked down PARP1, which assembles DNA repair complexes on chromatin that's been damaged. And we compared it to a PARP inhibitor, olaparib, that's used clinically. And what you can see is knocking down PARP in blue was more effective at controlling this triple-negative breast cancer. And the injections were given twice a week in all these models. The tumors that were treated with the PARP A6 had more activation of immune responses. Their CD8s to Treg ratio increased. Oops. They produced more, both the CD8s and the CD4 T cells in the tumors produced more cytokines, and actually knocking down PARP was much more effective at inhibiting PARP than the PARP inhibitor. Here we can show that the number of DNA damage foci increased, tunnel staining of DNA damage increased, and the results of unresolved DNA damage, interferon production, which increases tumor immunity, also increased. And when we compared PARP, and then we looked at still another mechanism, which is that tumors resist phagocytosis by antigen-presenting cells because they express a don't-eat-me signal called CD47. And Irv Weissman has pioneered therapeutics for CD47 with antibodies. So we knocked down CD47 with the EpCAM A6. And again, we had much better function and numbers of tumor-infiltrating lymphocytes, better antigen presentation. We had more M1 macrophages, which are the anti-tumor macrophages, versus the immunosuppressive macrophages. They expressed the molecules needed for antigen presentation. And they actually were able to show with the GFP tumor that the macrophages in the tumor were much more actively phagocytosing tumor cells and presenting antigens. And again, when we compared with the CD47 antibody, the tumor suppression was much better by the A6. The TIL function was better. and there were fewer myeloid suppressor cells in the tumor. And then we took advantage of one of the main properties of ASICs is that it's easy to combine them into cocktails, which are sort of at the fundamental basis of cancer therapy. And we looked at a cocktail of 4 ASICs, most of which I showed you here: nonsense-mediated decay enzyme, the don't eat me signal, MCL1, which is BCL2 family, anti-apoptotic molecule, and PARP1, and we combined them here into a cocktail. And although each of these individually worked, the cocktail worked much better. And this is shown with the individual mice. The increase in CD8 TILs and in the ratio of the active TILs to the suppressor ones was dramatically increased. And in this model, anti-PD-1 had no effect, significant effect, whereas the ASICs did. And we could combine them and get synergy. And basically what we found is that this cocktail was able to very strongly upregulate the genes that are important for T cell function, many of them, and also downregulate the genes that are immunosuppressive and suppress T cell activation in both for T cells and improve macrophage function. And then we were able to show that not only in xenografts, but in a metastatic model, which I'm not showing you, but also in a very aggressive, spontaneous, genetically engineered mouse model of cancer, of triple-negative breast cancer, which nobody has been able to, as far as I know, been able to show immunotherapy effective at controlling. In this very aggressive model where almost all mice develop tumors within a month of inducing the oncogene. A cocktail of the ASICs really dramatically improved tumor control and immune function in the tumor. So basically, I think this demonstrates sort of one of the, possibilities of in the future for developing RNAi therapeutics going beyond the liver, and in particular for harnessing RNAi for cancer immune therapy. And I'd like to stop there. Thank you.
Excellent. Thank you so much, Judy. So we have a couple of questions from delegates. We have just a minute or two to answer them. So I think that there's a theme that's kind of emerging amongst the questions, and I'm wondering if you could speak to this theme for a minute or two. One is on the theme of serum stability or chemical modification of either the aptamer piece or the siRNA piece of these ASIC conjugates. Do one or both parts need to be modified, and how deeply has that been explored?
Okay. Yeah, so what I showed you, our data, is really without chemical modifications. The aptamer and the siRNA parts of the sequence are modified with fluoropyrimidines, and that's all that we've done. And with those modifications, if we take the ASICs and incubate them with either mouse or human serum, we get stability, no decay that's evident for over a day. So that's enough to stabilize it in the serum. But I'm sure that our data could be improved by chemical modifications, especially the siRNA. I'm not sure I would try to modify the aptamer because that's already been selected for very high affinity, but maybe changing the linker, changing the aptamer, we just took, tried a couple of siRNAs and chose one. We didn't optimize even the sequence. So I think that's very auspicious.
Great. And so the second question that kind of builds on that theme is, or the second set of questions, this is a beautiful, elegant approach. What are the main barriers to broader clinical application? What has been holding back? What are the major barriers to further use of this technology, as far as you can tell?
Well, the main barrier is that is getting a company to really do the development that's needed and tested. I've been trying. I'm still in discussions with a company, but I don't know. Somehow, I guess I started trying when I first showed I could block HIV transmission a long time ago, and I couldn't get any interest because at that time it wasn't clear RNA therapeutics were going to work, and nobody had ever developed any topical microbicide as a therapy, so it was too risky. Now I think I'm ready, so if I'm hoping to be able to develop it.
And are the uncertainties related to sort of just the platform being still unfamiliar, or are there DMPK challenges with the siRNA aptamer conjugates?
Yeah, so we haven't really done, you know, I'm an academic lab, so I don't really do drug development in my lab. I just sort of like to examine some proof of concept type experiments. So we haven't done PK. It's really needed to develop it. I think as we talked about, the compound should be optimized, but I think with the kinds of safety and efficacy we're seeing in the mouse, I think it's very attractive.
Great. One other question. One of the delegates, Ahmed Abdullah, asks, did you see phagocytosis of healthy self cells, healthy epithelial cells or blood cells after CD47 knockdown?
We didn't look at that, but we didn't see any gross abnormality in blood counts or, you know, there's a disease called hemophagocytic histiocytosis, but there was clearly nothing like that. I don't think, it's hard to get tumor cells phagocytosed, and normal cells are not generally phagocytosed.
Well, thank you so much, Judy. It's amazing to see how much progress has been made in 20 years from really the basic biology to now multiple drugs approved. So yeah, thanks again. I'm going to hand back over to Anamika, who will wrap up this session, and we will see you in the chemistry session in about 10 minutes. Go ahead, Anamika.
Okay, thank you very much, John, and thank you very much, Judy, for a very interesting talk. And I think it's really amazing to see what has happened over the past 20 years and how versatile this approach is. So I really like that Judy showcased that.